Hello, welcome in this video lecture. We are continuing our chapter first, that is Introduction to Molecular Life of Cells or Human Being. So, continuing the point, that is bond present in biomolecules. So, we have studied different biomolecules and also studied various kinds of bonding in such case. So, whenever biomolecules are considered, there are two important class of bonds are developed. That is, first important types of bonds are strong bonds. Particularly, these strong bonds are the covalent bonds present in the compounds like peptide, glycosidic, and particularly phosphodiester type of compound that form their backbones, and also they are containing another type, that is weak bonds or weaker non-covalent bonds like hydrogen, ionic, and Vander Waals bond. Now, whenever such two types of bonds are present, no doubt covalent bonds are strong and they forms a conformed structure backbone of that particular biomolecules. Whenever the different examples of covalent bond present in the various biomolecules are considered, then these covalent bonds are always developed from sharing of electron, similar to chemical bond, that is sharing of electron takes place and holds the monomers of biomolecule together. Means strong covalent bonds in the biomolecule always hold the monomers of biomolecules together. They provide backbone, and such kind of backbone stabilize the structure. So according to this, covalent bonds are always stronger, which are present in various types of biomolecules. Now, which type of covalent bonds are present? The first important covalent bond is the peptide bonds. So, guess where we have studied peptide bonds. So, remember that peptide bonds are present in various types of proteins. That is, protein biomolecules are developed by amino acids as a monomeric unit, carboxylic acid group of first amino acid as a monomer combines with NH2 group of second amino acid and gives formation of peptide bond having repeated linkage of C double bond ONH, CONH, and such type of linkages are called as peptide linkages. So peptide bonds are the covalent bonds, strong bonds present in a protein as a biomolecule. Yes, you got it. Then second important strong covalent bond is the glucosidic bonds. This is also we have studied. The glucosidic bonds are present in various types of polysaccharides and carbohydrates. Polysaccharides and carbohydrates, and no doubt these polysaccharides and carbohydrates are developed from monosaccharide units and they bonded with the help of glucosidic type of bonding, that is bond between carbon single bond oxygen single bond carbon. So such bonding is called as glucosidic type of bonding. This is we have studied in various polysaccharide or carbohydrates, that is particularly various types of sugars, that is particularly disaccharide, trisaccharides, then different types of sugars like maltose, lactose, then sucrose. All these are containing glucosidic type of bonding. This is second important example of covalent bonds or strong bonds present in the biomolecules. Third important bond that is in the class of strong bonds are Phosphodiester bonds. So phosphodiester bonds means what? Whenever different types of nucleotides combines together with the help of phosphodiester type of bonding, that is phosphorus group PO3 minus oxygen is bonded with another ester type of bonding, then these nucleotides are combined so as to form the nucleic acid, and such type of bondings are called as phosphodiester type of bonding, phosphodiester bonds. So this is the third example of covalent bonds observed in biomolecules, and fourth one is the disulphide bond. The disulphide bonds are always takes place between sulfur atoms in the cysteine, alpha amino acid cysteine residues, helping to stabilize protein structure. Means cysteines, after formation of disulfide bonds, they are getting cysteine as a protein. So cysteine is helping stabilizing protein structure, that is disulfide bond. So these are the four different types of bonds present in the form of covalent bonds in various biomolecules: peptide bonds in proteins, glucosidic bonds in polysaccharide, phosphodiester bonds are present in nucleic acid, disulfide bonds are present again in a protein, that is, for example, sulfur atoms are present in a cysteine as alpha amino acid, and it combines together with the help of sulfur type of bonding. Then second type of bonding is the weak bonds present in the various biomolecules, and these weak bonds are, first one are, these are the non-covalent. Very good example of weak bonds are hydrogen bonds. These hydrogen bonds, no doubt how they are developed. Hydrogen bond is always developed in between heteroatoms like oxygen, nitrogen or sulfur, and hydrogen atom of same or other molecule or organisms. These bonds are called as hydrogen bonds. These are always weaker than covalent bonds. These are particularly non-covalent bonds, and they are more electronegative atom. Particularly such type of hydrogen bondings are present in DNA and protein. DNA आणि protein मध्ये आपल्याला कुठला bond दिसतो? Hydrogen bond. Then second type of weak bonds are ionic bonds. Now these bonds are formed between oppositely charged ions, such as between R groups of alpha amino acids in protein. They are easily disrupted by changing the pH. So second type of weak bonds which are observed in various alpha amino acids are ionic bonds. They are formed between charged atoms, that is between R groups present in amino acids in proteins, and they can easily disrupted by changing pH. So these are the ionic bonds present in a biomolecule. These are again weak bonds. Then very important third type of weak bonds observed in what? Particularly biomolecules is the van der Waals forces. Van der Waals forces, and these are again weak bonds. Short range of attraction occurs between molecules due to temporary and fluctuating charges. Though charge processes are developed in various biomolecules, they are fluctuating charges. They develop a weak bonding force of attraction between two oppositely charged species, and they play a significant role in a protein folding and molecular interaction. The role in protein folding. Most of the protein functions are carried out by these van der Waals forces, and they are considered as a weak bond developed in biomolecules. So, molecular interaction and protein folding are developed due to van der Waals forces, which is observed in various biomolecules. Then, next important bonding weak bond are the hydrophobic interaction. Hydrophobic. The word hydrophobic interaction indicates that they are water heating interaction. They does not combine with water. So these are the non-covalent interactions that cause non-polar molecules to cluster together in a watery environment. Whenever biomolecules are present in a watery environment, then hydrophobic interactions are developed in the form of non-covalent interactions, particularly in non-polar molecules. So such type of interactions are called as hydrophobic interactions. Cluster development takes place. And second is, they are major driving force in protein folding and formation of lipid bilayers. So hydrophobic interactions are weak bonds observed in various biomolecules like lipids and proteins. They play an important role in protein folding and formation of lipid bilayer. So these are particularly weak bonds which are observed in biomolecules, that is hydrogen bonding, ionic bonding, van der Waals forces, and hydrophobic interaction. So this is very simple types of bonding which are present in various biomolecules. You have to learn it properly. Okay, thank you.